Yes, Dharani, very good evening. Mercy, very good evening. Yes, Vivek, hello. Very good evening, Vivekanandan. Yes, happy evening to you all. So today we are going to discuss photosynthesis. Yes, Tarunika, very good evening to you. Yes, as Mercy and Dharani are there, let me start the class with a small joke. Let me see how Dharani or Mercy or Tarunika cracking this joke. Okay. Can you find out which is the terrible vegetable? Very terror. Terror vegetable it is. Which is the terror vegetable? Yes, Latika. Very good evening, Latika. Very good evening. So, which is the terror vegetable, Latika? Let me see who is finding the answer. Is it Dharani or Mercy or Vivek or Tarunika or Latika? Yes. Which is the terror vegetable, my dear Mercy? Have you found out? Shall I reveal the answer? Yes. Yes, ma. Which is the terror vegetable? So you'll be afraid it will beat you. It will beat you. Drumstick. Okay, Tarunika, yes, it is drumstick. You may be. Yes, one such answer. Excellent answer, Tarunika. No idea, Dharani. Yes, it is beetroot. Yes, Tarunika said drumstick. That also might be the answer. Yes. Yes, my dear Mercy. Drumstick. Yes, you will be afraid of the sticks, na? Yes. So these three can be a terror vegetable. This question exclusively for Dharini and Mercy. Yes. Shall we proceed into the photosynthesis? Can you answer me, children? What is photosynthesis? What is photosynthesis? Yes, nice question, na Mercy. Yes. So what is photosynthesis? Using sunlight. So you can see in the picture, light energy is there. Of course, carbon dioxide. Oh, okay. Yes. Carbon dioxide, water, all these together we prepare glucose C6H12O6 and release oxygen. Of course, this is what photosynthesis is happening. Can you see water, carbon dioxide and light energy? Of course, oxygen is coming out and we prepare carbohydrates. Okay. So, carbon dioxide will be in the atmosphere. So, why the photosynthesis has to happen? Yes, Sachin, very good evening. Why photosynthesis has to happen? Why it is necessary? So you will be having in between questions on each and every paragraph. This time I prepared. Hi Pushpa Kumar, I missed you a lot. How are you? Very good evening, happy evening Pushpa Kumar. Hope to see you early. I am so fine. Missed you a lot. So tell me, why plant has to do photosynthesis? Yes, very good. To prepare energy as well as of course food. Food is prepared only by plant, then it can be distributed to animals. So it may be a carnivore even, but the herbivore get the food only from the plant, yes or not? Okay. Yes, sir. PK big fan, yes, sir. For you also. So let us discuss what are the experiments or with reference to the photosynthesis. Okay. See, this is the experiment, of course, where you can find variegated leaf. Some portion is having green, other portion may not have the green. Okay. So when you do the starch test with the this particular leaf, that is when we add iodine, we can get blue black wherever green color is present. So where other colors are present, we may not get blue black color. So it indicates what you can confirm from the starch tester from this variegated leaf. Yes, Tarunika also telling hello Pushpa to you. Yes. So come soon. Let us see. Yes. So what do we infer from this experiment? Wherever we have the chlorophyll, there only photosynthesis might have happened, starch might have been prepared. So chlorophyll is essential for photosynthesis from this experiment. Yes, sir? Now next experiment, if you see, this is called Mohel soft leaf experiment. Okay. So here you can take a bottle with potassium hydroxide and half of the leaf is inserted into the bottle. The other half we are not inserting. It remains outside. After some time when you do the starch test again, here it turns blue-black, whereas where we expose to the potassium hydroxide, it is not forming blue-black. Yes, sir. So it indicates what? What we can infer from this experiment. Yes, my dear children, can anybody answer me? Your time starts now. Yes, 5. For food, here alone it turns blue-black. Where we expose to potassium hydroxide, it is not turning blue-black. So why? What you can confer from this, Mercy? 4. The time starts. Very good. CO2 is required for the photosynthesis. Excellent, Dharani. You're rocking. So CO2 is required. When you expose the leaf to the potassium hydroxide, CO2 is absorbed. Without CO2, we cannot make carbohydrate or glucose. Yes, sir? Very correct. Excellent, Dharani. 100% correct. Now, can you tell me, this is the question, the off-leaf experiment where a part of the leaf is enclosed in a test tube containing some potassium hydroxide soaked cotton was performed, which shows that dash was required for photosynthesis. You can answer me now. Yes, your time starts. What is required? Yes, Dharni said C. Anybody else? Yes, Latika, it is CO2 and Dharni also answered correctly. Yes, it is CO2. Like this, only questions will be asked from historical background. So, every historical background, I also framed a question. Let me see this time. Yes, Parthasarthi, very good evening. Very good evening. Yes, C is the correct answer. Yes, Ajay, it is CO2 for this question. Okay, shall we proceed to the next? So, this is performed by which scientist? Joseph. Yes, Sarnika, it is CO2. Excellent answering. So, Joseph Priestley in 1772. So, it's a storyline only. So, when he kept a bell jar and a rat, a candle was kept. The candle blow off. And he kept a rat even. That also did. But when he keep a plant over there for prolonged duration. Yes, it is Presley. Very correct, Bhartasarthi. It is Presley in 1772. Of course, after prolonged duration in the same bell jar, he also introduced a candle as well as a rat. Both survived. So when the mint plant actually, so he was using what? Mint plant. And what he concluded, the foul air is purified by the plant. So the foul air is purified by the plant. This is the conclusion of Joseph Priestley. Yes. Shall we see the question what is coming now? So what is incorrect with reference to the scientists and their usage of the plant? Priestley used neem plant, John Ingenhouse's aquatic plant, Engelmann cladophora, and Von Neil purple and green bacteria. Yes, your time started now. 5, 4, 3. Which was the wrong combination? You are supposed to find out. 2 and 1. Yes, it is option A. Joseph Priestley was using what plant? Yes, my dear Latika, very correct. Dharani and Pathasarthi and Tarunika, excellent answer by all of you. Can you tell me what was the plant Joseph Priestley was using? What was the plant he was using? So this is by John Ingenhouse. Okay. So in 1779, so 1779, this experiment was performed and he was using some green algae plants. Very good, Dharini, and very good, Latika. It is mint plant. Excellent answer by both of you. Yes, it is mint plant, 100% correct. Okay. So, John Ingenhouse, what he was doing, he was using some hydrophytic plant. You can see the setup with a funnel, and he kept the test tube over it. This setup, he kept it in the light, another one he kept it in the dark. The same setup, one in the presence of light, another one in the absence of light, dark condition. So when it was in the light condition, you could able to see the bubbles can able to form. So you can found the bubbles in the light condition. Whereas in dark condition, the bubbles are not formed. So what he conferred, the foul air is purified only in the presence of light. Okay. So foul air is purified only in the presence of light. So that is the conclusion given by John Ingenhouse. Based on the conclusion, we will be asked a question. Okay, children, shall we proceed? Now, next one is, of course, it is Julius Van Shaw. Can anybody tell me what was his conclusion, Julius Van Shaw? Yes, Parthasarthi, Latika, Tarunika, Dharani, Vivekanandan, Ajay. Anybody? Julius Van Shaw? Yes, he said the plant prepare glucose and reserve it in the form of starch. Okay, so this was the experiment related to the Julius Van Shaw. Of course, he didn't give the experimental chemical formula and all for this, but he is the one first said, of course, glucose can be synthesized with the help of the sunlight and all that. But he said that now this is a question with reference to Julius Van Shaw experiment. So what is it? Production of glucose when plants grow, location of green substance in plants in special bodies. Which of the following will be true? Glucose produced during photosynthesis is usually stored as cellulose in plants. The green substance refers to chlorophyll. Special bodies refers to chloroplast. So among the three ABC points, which are the points correct? Your time starts now. Yes, Dharani, Tarunika, Parthasarathi, very important question you can attend. Either you can write 1, 2, 3, 4 also. Your time started now. 5, 4, 3, Tarunika answered 2. Anyway, let us wait for others also. Dharmi also answered 2. Yes, it is 2 actually correct. B and C are the correct statement. So glucose are produced during photosynthesis. Is usually stored as starch, as you all know. It is not stored as cellulose. The green substance, of course, it may be chlorophyll A and B, and special bodies are chloroplasts. So B and C is the correct answer. Excellent. Yes, Tarunika as well as Parthasarathi and Dharini. Excellent answer. 100% you are all correct. You are all rocking. Okay, like this only each one we should try one question, then it will be easy for us to remember all the critical points of that particular paragraph. Now this is in 1854 Engelmann experiment. So what he was using? He was using sunlight. The sunlight is passed over the prism. Okay. And you will get spectra of light here. Vibgayar, you know. And he was using a bacteria as well as the algae. The algae name, can anybody guess it? What is the name of the algae? Yes, Dharani, yes. Latika, Parthasarathi. It starts with C. Yes, it is cladophora. Excellent, Dharani. You are rocking today. And Latika, cladophora is the correct answer. Excellent answer. Yes, it is cladophora. Of course. So, cladophora is a green algae. And you can find the bacteria also here. The bacteria are aerobic or anaerobic. The bacteria what he used for the experiments, is it aerobic or anaerobic? Yes, it is cladophora. The bacteria, I am having only two options, either aerobic or anaerobic. Or anaerobic. Yes, before I complete the answer, yes. It is aerobic bacteria. Very correct. Excellent. Latika and Dharini. Very good. Keep it up. So, it is actually aerobic bacteria he was using. And using cladophora, these aerobic bacteria produces oxygen. And these oxygen are accumulate more in the blue light as well as on the red light. Okay. So 400 and 680 nanometer they were more. So absorption is maximum. Photosynthesis is maximum in blue and red light. Studied by Engelmann in 1854. Okay. So this is his experiment. Yes, Tarunika. The action spectra of photosynthesis. Action spectra. Studied by whom? Is the question. From the previous paragraph. Can anybody guess it? Yes, Parthasarathi. Who is the action spectra of photosynthesis studied by? Yes, it is single man. Excellent. Of course, you know the other possible questions are what was the algae he was using? Cladophora. What was the bacteria he was using? Aerobic bacteria. Which are the two lights, blue and red? These are possible questions. Yes, it is single man. Dharani as well as Parthasarathi and Tarunika. Excellent answering by all of you. Okay. So this is another very long question related to Engelmann experiment. You are going to find out how many statements are correct. Please try. I'll read anyway. In Engelmann experiment, the aerobic bacteria accumulate mainly in the region of green light. Action spectra of photosynthesis corresponds closely to absorption spectra of chlorophyll A in chief photosynthetic pigment. So chlorophyll A is the chief photosynthetic pigment. Some organisms do not release oxygen during photosynthesis. There occurs a clear division of labor with respect to light reaction and dark reaction within the chloroplast. Okay. Purple and green sulfur bacteria can liberate oxygen during photosynthesis. So, how many are correct? How many are correct? You are going to answer me. Yes, my dear children. Dharni answered 3. Parthasarthi answered 3. Yes, 3 is the correct answer. Very good. Excellent answer by you all. Of course, it is 3. So, these are the statement. Of course, chlorophyll is a chief pigment. Green bacteria and all does not, green sulfur bacteria does not release oxygen. You have light reaction and dark reaction. So, these 3 are the correct. Green sulfur bacteria release sulfates. So, this is wrong and this also wrong. So, these are the correct answer. So, it is not 2. Yes, it is 3. Very correct, Arunika. Excellent answer by you. Yes. And this is the reaction given by von Neer. Okay, in 1884. So, you know, this is the basic reaction using carbon dioxide and water. Light carbohydrates is prepared and oxygen is released. And of course, in case of green sulfur. Vanga Sanjay. Hello, madam. Very good evening. Yes, Ajay, you're there. Green sulfur and purple sulfur. We were discussing so many questions. You were not there. So, இப்போ வந்து the purple sulfur and green sulfur bacteria this A. So, only the A is released, which is H2S actually. So, sulfur will be released. So, he said oxygen is released. But do you know who proved that oxygen is released from carbon dioxide? Any of you remember? Actually, 2 scientists it is. Yes. Why you are worried about Tarunika now? Yes, tell me. So, who proved that oxygen is coming only from water, not from carbon dioxide? Anybody? Yes, my dear. Yes, Latika. Larni. The name starts with R. Okay, shall I write? It is Reuben and common. Reuben and common. Yes, Latika, very correct. Excellent. Reuben and common are the two scientists. They were using radioactive isotope O18 actually to prove that oxygen is coming only from the water, not from carbon dioxide. Very common question. Definitely you will come across when you are practicing. So Reuben and common are the one who experimentally proved oxygen is coming only from water, not from carbon dioxide. And now they proved using radioactive isotope. What was that radioactive isotope? O18. These are the possible question, of course, here. Shall we proceed to the next one? Yes, it is a question, my dear Sanjay. You can answer me now. By studying purple sulfur bacteria and green sulfur bacteria, who was the first scientist demonstrate 1931? It is light dependent redox oxidation and reduction reaction in which hydrogen from oxidizable compound reduced to carbon dioxide. Yes, it is 1931. Your time started. Yes, Ajay. Now, Ajay did some detective work. Shall I ask you one question? Which was a detective vegetable? Which was the detective vegetable? Dharmi said answer. Yes, Tarunika also answered. Which is the detective vegetable also? You can answer Dharmi now. Von meal is the correct answer. Yes, Latika, very correct. Excellent. Yes, Ajay. Who is detective vegetable? He will have a black eye. That is the clue. Black eye. Coconut. Okay, might be. Dharmi answered one. But black of eye we used to learn. Let me see. Anybody else is thinking? Yes, my dear Sanjay. Think well. Parthas are they? Anybody? It has black of eye. No, vegetable itself is detective. Yes, Parthas are the answer. It is potato. Potato, we used to say no black of eye for the rejuvenation of plant. Yes, sir. See, Sanjay even gave a potato. Yes, that is detective vegetable. Fine. So before we proceed to the photosynthesis site and pigments, are you all clear about historical backup? What and all we discussed, Von Neil and all, and we have to discuss regarding immersion also. See what he did, immersion, the same action spectra you can take and light is passed with Gaia, violet, indigo, red and all. Okay, now he is giving the light beyond red light. Light is used beyond red light. Beyond the red means it may be infrared, ultraviolet. Okay. Now what will happen to the photosynthesis? Will it increase, decrease, not happen? What is your answer? So immersion did this. Potato we gave answer and now only you are thinking about coconut. Potato, black of eye. Coconut also is having eye, you can say. Yes, Ajay. When you say I will accept Ajay. Now tell me. In dictionary where we could find more B. What Ajay? Where we find more B? I didn't get you. Answer me. Any answer? Beyond red light, will photosynthesis happen? What are so much girlfriends and all for you, Ajay? So beyond red light, will it happen or not photosynthesis? Yes, Sanjay is asking. Beehive. Okay. Yes, Sanjay, you're rocking today. You're my student. You're telling another mocha joke to me. Yes, thank you. Now, what happened actually? The photosynthesis will reduce. Yes, it won't happen. So, it won't happen is the correct answer. Beyond red light, it will drop. So, only it is called immersion red drop method. So, that was studied by immersion. Again, it is not there in NCRT, but possible question. Yes, sir. So we have to discuss the site of photosynthesis, which you all know. Leaf is the kitchen of plant. If you take the leaf, where you find in cross section epidermis. Between epidermis, we have mesophyll. Okay. And mesophyll, we have palisade parenchyma, where you find chloroplast. And these chloroplast is having outer membrane, inner membrane, stroma, grana. So you know what is grana. So you have one thylakoid. This is thylakoid. Yes, epidermis is also there. Upper epidermis and lower epidermis. Between that you are having mesophyll. The thylakoid, if they are in compile, they are considered to be grana. Yes. Spongy too we have. That will have air only, does not have chloroplast. Only palisade we have chloroplast. Yes or not, Sanjay? Which is the hall of plant? Tirilier. What is that, Ajay? See, all are my student, okay? So tell me, what is that hall of plant? Hall of the plant is what? What ra Sanjay? Find ra. No, Okay, Ajay will answer. So grana is there. And you can find actually the grana with stroma lamellae, connected by stroma lamellae. And this is stroma or matrix. Spongy parenchyma will have air cavities. So they are for exchange of air. So it is called parenchyma. We have chlorophyll only, that is chloroplast only in the palisade parenchyma. That mostly restricted in upper epidermis. In certain cases like nerium and all, you find multiple epidermis. So those cases you have palisade parenchyma even here. So spongy parenchyma does not have chloroplast. Okay, Sanjay. Yes, sir. Cleared up. Shall we proceed? Yes. Okay. Fine. So this is the condition actually we can find. And you know there are light reaction and dark reaction. So before we proceed into the light reaction and dark reaction, can you tell me where the light reaction will be carried out and where dark reaction will be carried out? Imbibe, they said spongy parenchyma, but generally NCRT does not say that. We should follow imbibe or NCRT, my dear Sanjay. And imbibe does not answer everything correctly, right? And you should not use imbibe here in social media. Yes, dark reaction stroma. Okay, so dark reaction we have in stroma. Light reaction where it will be, of course, in thylakoid membrane. Okay, light reaction in thylakoid membrane as well as, yes, of course, in thylakoid membrane or grana fine. Apart from that, one more place light reaction will be carried out where it is. Very good. Stroma lamellae. Yes, Latika, it is stroma lamellae. Very correct answer. Excellent it is. Okay. And of course, dark reaction, you all said stroma. So, light reaction, what and all we prepare? We synthesize what in light reaction? Yes, lamellae is correct. And what do we synthesize in light reaction, my dear children? Yes, Dharani, excellent, 100% correct. We synthesize ATP and NADPH. So, ATP and NADPH we prepare in light reaction, and dark reaction we synthesize, of course, glucose. Glucose is synthesized in dark reaction. Okay. So, you know how the light reaction and dark reactions are going to happen. Before that, let me share a small story to motivate you. Shall I? Are you ready to listen the story? Yes, Dharani and Latika, Sanjay, Ajay and Bhartasarthi, everyone. And Tarnika, yes. Okay. The story is nothing but is a movie. Many of you might have watched Kunjan Saxena. Have you watched Kunjan Saxena? Your favorite Janvi Kapoor acted, Sanjay? You watched. Very good. So, in that story only I am going to share. Okay. You're ready. Fine. Yes. So, அதில் வந்து ஜான்வி கபூர் தான் நடிச்சிருக்காங்க. இட் இஸ் அ டாக்குமெண்ட்ரி மூவி. அந்த மூவில வந்து அவங்க பேரண்ட் எல்லாம் வந்து ஆக்சுவலி மிலிட்ரியில தான் இருந்திருப்பாங்க. ஃபைன். அண்ட் ஷீ ஆல்சோ வாண்டட் டு பிகம், எஸ், ஸ்ரீதேவிஸ் டாட்டர். She wanted to become a pilot officer. So she tries. அப்ப ஃபர்ஸ்ட் அட்டெம்ப்ட் பண்ணும்போது அவங்க வந்து கொஞ்சம் வெயிட்டா இருப்பாங்க, அவங்கள ரிஜெக்ட் பண்ணிடுவாங்க. She didn't give up. Again she tries. When she tries for the second time, நிறைய rounds அதுல காம்படிஷன் எல்லாம் இருக்கும், அதுல உங்களுக்கு தெரியும். She crosses everything. அதுல 4 பேர் ஃபில்டர் ஆகுறாங்க கடைசியா. அந்த 4 பேர்ல 2 பேர் வந்து செலக்ட் பண்ண போறாங்க. 2 பேர் of course they have to get rejected. அதுல ஒரு கேர்ள் வந்து நீங்க பாத்தீங்கன்னா she may be in the top scorer மாதிரி 9, 9.5, 9.8ல இருக்காங்க அந்த அம்மா 10க்கு எல்லாத்துலயும். So she'll be damn sure selected. அப்ப இந்த 3 பேர்ல யாரோ ஒருத்தருக்கு ஒண்ணு கிடைக்க போறது அப்படின்னு இவங்க டிசைட் பண்ணிட்டு இருக்காங்க. இப்ப the final testing has been carried out. இப்ப அவங்க என்ன பண்றாங்க, they are taking them to the 4th floor. So 4வது மாடிக்கு அந்த சீஃப் ஆபீசர் வந்து அவங்க எல்லாரையும் கூட்டிட்டு போறாரு. அந்த மாடியில கூட்டிட்டு போயிட்டு அவங்க எல்லாரையும் அந்த ரூம்ல பாத்தீங்கன்னா ஒரு பெரிய விண்டோ இருக்கு, ஒரு ஆள் நிக்கிற அளவுக்கு. அந்த விண்டோ மேல ஃபர்ஸ்ட் அந்த பொண்ணு 9.8 எல்லாம் சொன்னேன்ல, அந்த பொண்ணு தான் போறா. போனதும் அவர் சொல்றாரு கிளைம் ஆன் த விண்டோ அப்படின்னு சொல்றாரு. ஏறிடுறா. ஏறிட்ட பிறகு அவர் என்ன சொல்றாரு, ஜம்ப். ஜம்ப்னு சொன்னதும் அந்த அம்மா என்ன பண்றது தெரியல, கீழே குனிஞ்சு பாக்குது, அவ்வளவுதான், விழுந்தா ஒரு எலும்பு கூட தேராது. 4வது மாடி அந்த மாதிரி இருக்கு. சோ அது யோசிக்குது, நம்ம இப்படிலாம் ரிஸ்க் எடுக்கணுமா அப்படின்னு சொல்லிட்டு அந்த அம்மா என்ன பண்ணுது, இறங்கி எனக்கு உங்க வேலையை வேணாம் போ அப்படின்னு சொல்லிட்டு போயிடுது. யாரு? இந்த டாப் ஸ்கோரர். சோ நம்மலாம் டாப்பர் அவங்க தான் வருவாங்க அப்படிலாம் யோசிப்போம் தெரியுமா? நம்ம மனசுக்குள்ள அவங்கலாம் நல்லா படிப்பாங்க நமக்கெல்லாம் எப்படி? அப்படிலாம் யோசிப்போம் பாருங்க. அந்த டாப்பர் என்ன பண்ணிட்டாங்க? கீழ குதிக்க முடியாதுன்னு சொல்லிட்டு போயிட்டாங்க. இப்போ இந்த அம்மா போறது யாரு? கொஞ்சம் சக்சேனா. யுவர் ஃபேவரட் ஜான்வி கபூர். சோ இந்த அம்மா வந்து அதே அவர் என்ன சொல்றாரு ஆபீசரு? கிளைம். கிளைம் பண்ணிடுது. நெக்ஸ்ட் என்ன சொல்வாரு? ஜம்ப். குனிஞ்சு பாக்குது. பார்த்ததும் எப்படி இருக்கு? அவ்வளவுதான் ஒழுங்கா ஒரு எலும்பு தேராது. இதுவும் அதே தானே யோசிக்கும். பாக்குறது. ஆஹா ரொம்ப ஆழமா இருக்குதே என்ன பண்றதுன்னு. டைம் போயிட்டே இருக்கு. அந்த டைம் ஷெட்யூலிங் டைம். அது யோசிச்ச பிறகு என்ன பண்ணுது? ஒரு டர்ன் பண்ணிட்டு she jump inside strategy. ஓகேயா? சோ டாப்பர் தான் வருவாங்கன்னு நீங்க யாருமே யோசிக்க கூடாதுன்றதுக்காக தான் இந்த மோட்டிவேஷன் ஆக்சுவலி. யுவர் ஸ்ட்ராட்டஜி அண்ட் ஸ்மார்ட்னஸ் இஸ் வேலிட். அந்த இடத்துல she had a spark. She jumped inside the room. He said only jump. He didn't say that you should jump down. அப்படிலாம் ஒரு ஒண்ணுமே சொல்லலையே. ஸோ ஸ்ட்ராட்டஜி நமக்கு எது ஈஸியா வருமோ அந்த ஸ்ட்ராட்டஜியை நம்ம அப்ளை பண்ணணும். ஐ ஹோப் எவ்ரிபடி கேன் ஸ்கோர் மோர் இன் பயாலஜி. ஸோ அது உங்களோட ப்ரையார்மோஸ்ட் ஸ்ட்ராட்டஜியா இருக்கணும். அண்ட் நான் எப்பவுமே சொல்ற மாதிரி பயாலஜி திரும்ப திரும்ப நம்ம படிக்க வேண்டி இருக்கும். பிகாஸ் அந்த டேர்ம்ஸ் எல்லாம் நம்ம ஞாபகம் வச்சுக்கணும். டயக்ராம் கூட வரைஞ்சி லேபிளிங் பண்ணி பார்த்துறதுனால ஒரு தப்பும் கிடையாது. அண்ட் டைம் டேபிள் நான் எப்பவுமே சொல்லுவேன், நீங்க டைம் டேபிள் போட்டுட்டு அந்த டைம் டேபிள ஸ்ட்ரிக்டா படிக்கிறதுனால உங்களுக்கு வந்து ஃபாலோ பண்ணீங்கன்னா your score will rise. Same way you can follow for chemistry and physics also, my dear children. So this is a small motivation from my side. If you really like the story, please comment. Also give me one like. Thank you. Yes, Dharni. Thank you. So even put it in the comment section about the motivational story so that many will like and try to follow every week. என்னால முடிஞ்சது உங்களை நான் மோட்டிவேட் பண்றேன். I know you also may have little stress, exams, pressure எல்லாம் இருக்கும். அது என்னால முடிஞ்ச அளவுக்கு I will be reducing it. Okay. Yes, Sanjay. So what are the pigments involved in photosynthesis? We have chlorophyll. Chlorophyll A, B, C, D and E. இதெல்லாமே வந்து நமக்கு pigments தான். இதுல which is the prime pigments? Anybody answer me? Either it is A or B or C or D or E. Which is the prime pigments? Tarnika, is it A or B? Okay, Sanjay also answered B. Actually it is A. Yes, my dear Gopika, my dear child, very good evening. We are almost at the end part. Anyway, so you are coming to the climax scene. Like the policeman entry in Tamil movie. Gopika has come to rescue us. Anyway, thank you Gopika for your presence. So, chlorophyll A is the prime pigment. Okay. And what is the formula for the chlorophyll A? It is C55 H72 O5 N4 and magnesium. Okay. So we have, what is the functional group here? We have functional group methyl in chlorophyll A. What is the color of chlorophyll A in incident light? Possible question. It is bluish green. In transmitted light, so this is in incident light, all these are possible question. Incident light, it is bluish green, and transmitted light, we have red color. Okay. Now, if you take chlorophyll B, so this is not a prime pigment anyway. The formula is C55, H70, O5, nitrogen 4 and magnesium. So here the functional group is CHO. The color is olive green in incident light, and transmitted light it is brownish red. Okay, so this is the pigments actually. And if you see the structure of those pigments, you can find a magnesium in the middle with 4 pital ring, tetrapital ring they used to say head. Of course, these structure and all they are not asking, but sometimes they are asking what is in the main mineral you can find in the middle. Of course, it is magnesium as you know, and you can find 4 nitrogen and 20 carbon will be in the tail. 20 carbons you can find in the tail region. So remaining of course 35 carbon will be in the porphyrin head. So this is porphyrin head. And this is phytol tail. Okay. And if you see the other pigments, we have carotenoids also. And carotenoid, what is the formula? C40 and H50. 55 and 56. Okay, so this is regarding carotenoids. And then we have picobilin also. So if you see these are pigment molecule. What is the other name we can give for pigment molecule, children? Any of you know? The answer even present in the board. What is the other name of pigment molecule? They take the light. They absorb the light. They absorb the light. What is the other name of pigment molecule? Anybody? The answer is in the board itself. It is light harvesting complex, my dear children. It is light harvesting complex. Okay. And you know, they transfer the energy to the pigments. Like, first of all, if you see picobilins, they transfer to carotenoids. Then carotenoids are transferring to chlorophyll. That is B or C, whatever it is. Then they transfer to the reaction center where we find chlorophyll A alone. So transferring to reaction center. From there, of course, the electron will be, light energy will be converted into electrons. Of course, it will be transported. Yes, sir? Yes, it is light harvesting complex, what I asked. The other name for the pigmented collection. Yes, sir? We used to say even antennae complex. So reaction center is where we find chlorophyll A only. So based on this light absorption, we classify how many photosystem, children? How many photosystem we classify? How many photosystem we classify? Is it 2 or 3 or 4 or 5? Yes, my dear Dharani. Dharani said 2. Parthasarthi. Yes, it is 2. Of course, you know photosystem 1 and photosystem 2. We have 2 photosystem 1 and 2. So what is the absorption spectra of photosystem 1? Any answer? Yes, Shalini Prabhu. So yes, Gopika, you can be happy. Shalini came just now. Okay. So photosystem 1, what is the absorption spectra? Is it 680 or 620 or 480? What is that absorption spectra of photosystem 1? Yes. Yes, it is 2. We have 2 photosystem. What is the absorption spectra of PS1 and PS2? Any guess? Parthasarthi, Dharni, PS1. Okay. This is PS1, PS2. Yes, my dear Sanjay, Latika. So PS1 you all wrote. What about PS2? Yes, Gopika answered. It is 680 for PS. Anybody else would like to answer? Yes. It is 700 for PS1 and 680 for PS2. So 700 and 680. Okay. So we didn't discuss the absorption spectra. Of course, can anybody try with the graph which is X and Y, Z? You can get green color peak X, Y and Z with the absorption spectra. Can you guess it? Yes. Is it 3 or 4, Taranika? 3 or 4? Little deviation between 3 and 4. Little deviation only between 3 and 4. See, X is chlorophyll A. You can find 2 peaks, of course. Y is closer to peaks, that is green and yellow, which is carotenoids. And Z you can find 2 peaks closer to the green, which is chlorophyll B. So A carotenoid and B is coming in answer 4. So 4 is the correct answer. Yes, Arunika, Parthasarthi, Dharani and Latika excellently all answered. And many might be watching cricket, I know. Anyway, thank you all for participating. In next class, we may be discussing light reaction and dark reaction. Thank you so much for your valuable time. I hope you all understood. Any doubt, please ask me and leave. Otherwise, we can wind the class for today. Thank you, children. Thank you, Tarnika. Thank you, Latika. Thank you, Gopika. Thank you, Dharni. Thank you, all children.